Why what?
Why is the red all at the top? Why is it polarized?
Where is the red to the
Why
top?
is the red polarized to the top of the mitochondria, right? The Z line's in the middle running up, is that right?
This Z line here?
Yeah. And see the red is distributed?
Is here means?
See the red is distributed like a cat? Or is that just my eye? Can I have the pointer? I'll point to
Yes.
See, it's distributed at the top.
Oh, yeah. We don't know that yet. We don't have the high enough resolution and particularly look at the time-lapse microscopy to see what happened in the next few minutes or something. What we notice is that these red puncta always kind of set apart from the main mitochondrial network. So your question could be related to this: is this part going to be budding off as a mitofission process? We don't know that. We don't know the answer. And we don't have the time-lapse microscopy at the resolution high enough for us to dissect that question.
Looks like they got the wrong one.
Can the Barshop hear me yet?
This is Texas Biomed.
There's 4 sites that are up. I just spoke to the tech.
We keep going. And so when we did the co-staining of LAMP1 and the Mito Timer, we found this lysosome marker always co-localized with the red puncta. So this suggests that this red puncta comes from mitochondria, and it's targeted by lysosome. And that reminds us of this property of lysosome. Lysosome has a lower pH, and it's a very acidic. When the acidic lysosome fuses with the autophagosome containing mitochondria, this acidic environment kills GFP signal. So the GFP signal is very sensitive to acidic environment. So this is quite possible that this is due to the mitophagy event with the lysosome targeting, suggesting this could be used as an indication of active mitophagy. So we test the question whether this change in the ratio in this green and the red is really sensitive to oxidative stress. So what we did is we treat the cells transfected with a Mito Timer with the actin-myosin A, rotentin, and the paraquat. These 3 cause mitochondrial stress. We know that. And when we measure the ratio of each cells and count many, many cells in the same plate, and we conduct these many experiments, and we found that this ratio increased with these 3 treatments. That's in cell culture. The fly heart. We feed the fly heart with 3 drugs, and it changed the ratio, as you can see here. This is a control. Increase the ratio, increase the ratio. Rotentin has to be fed in a different way. So we use a different control, and you can see the increase in the ratio. And also, there's a trend of increase the number of red puncta in the heart after feeding those flies with antimyosin A, paraquat for 24 hours or 4 days with the rotentin. So suggesting that if we artificially increase the oxidative stress, we can see the changes in those. But the red puncta increase could mean increased autophagy, mitophagy. Aging. This is a 70-day fly versus 7-day fly. You can see the difference here, and we quantify the ratio and quantify the number of red puncta in the heart. So we went on to ask the question, can exercise improve it? Can exercise improve autophagy and mitophagy? And what happened to an acute bout of exercise? So actually, to our surprise, we were expecting that immediately after 90 minutes of treadmill running, and this is a quite intense exercise, we would expect in the muscle that we would see increased mitochondrial, the red to green ratio. We didn't see that. We saw that peak at 6 hours post-exercise, and that decreased to the basal level 24 hours after. So this is like a transient increase in we call it mitochondrial stress. and the mitophagy. So this is the total ratio, and also we quantify the dots, as you can see here. I'm convinced this is not an artificial experimental artifact, because we inject the contralateral muscle with another timer, we call it ER timer. We targeted this to ER, and we expect to see similar changes. We didn't see any changes in the ER timer injected muscle and used as a control. So this was disappointing for us to not see any kind of so-called ER stress or ER oxidative stress, but that validates the finding in the other leg. So we quantify that. Yes? The construct injected is the same timer protein, but it's not a targeting to mitochondria, but targeting to ER.
And that's injected into the muscle, or it's a...
It's injected into the other contralateral leg. So this mouse, using 2 legs to exercise, we quantify 1 leg with a Mito Timer, ER timer with the other leg. It's electroporated, yeah. It's 10 days after electroporation. Let them totally recover from the electroporation and then exercise them. So this is the quantification of the ratio of the Mito Timer versus the number of red puncta. Here is the ER timer. Only here showed a few dots here increased, and we don't know what that means. But ER environment seems to me is a quite stable environment. They can tolerate exercise quite well. So with this technology, I think we can quantify mitochondrial structure, content, possibly the oxidative stress, history of oxidative stress, mitophagy. Here's an example. This is the exercise training. We put mouse in the running cage for 6 weeks. After 6 weeks, we took those muscles and measure the size of the mitochondria. So this is a percentage of mitochondria occupied space here. There's an increase from 35% to 40%. There's a moderate decrease in the ratio, suggesting they are healthier, they are more green. And then here's the red puncta. It didn't show much of a change. So suggesting that exercise training can improve mitochondrial quality, increase the mitochondrial content. But if you notice that the size of mitochondria are bigger, and one of the things is that mitochondria become like a uniform, and the 2 mitochondria on each side of this almost equal size. When you go to the control muscle, and mitochondrial size are quite different. There is a discrepancy or uniformity improved. So,
Can I ask you a question?
yes?
So the driver you're using here is a...
Constitutively active promoter. But electroporation only transfect adult myofibers. No other cells will take up the plasma DNA.
If you had increased production of the timer under exercise, the newly formed timer, Mito Timer, is green, right?
That's correct. In this case, it's not the case because the promoter is a viral promoter. It's not influenced by exercise training.
It's not influenced by. So
No.
you know that there's not a change in your
No. Yeah, so here, what we did, it was scan this intensity of different mitochondria here and overlay on top of this. This is a sedentary muscle. As you can see, the peak not always go together, right? Some peaks here big, some smaller. Some has a peak here, but you don't have a peak here. You don't have a peak here, you don't have a red peak here. But here, it always goes together. So suggesting that improved structure of the mitochondrial reticulum. This is quite similar to published data from the Japanese group many years ago using scanning microscopy and transmission electron microscopy. They show that more robust mitochondria in the slow twitch muscle, less uniform mitochondrial network in the glycolytic muscle, so they draw these cartoons here. And if you look at our Mito Timer structure, it is quite similar here. This is the slow twitch muscle, have big mitochondria here on each side of the Z line. Here is the intermediate one. This is the glycolytic muscle. So our next question is, can this be used to detect any mitochondrial stress, oxidative stress that are known to us? So what we have done is we have this normal child mouse data here, and then we fed some of the mice with high-fat diet for 10 weeks. As you can see, the structural change is huge, and you have this enlarged red puncture here, the accumulation of the red Mito Timer signal, and so this is it's an association at the same time with insulin resistance. So if you feed those mice with high-fat diet, you saw those changes, and we used the micro-PET imaging and stimulate animal with insulin and look at the FDG uptake in the hind limb. In the mouse with a normal chow fat and versus the high-fat fed animals, you can see this reduction in insulin-stimulated glucose uptake. It's more like insulin sensitivity, direct measurement of insulin sensitivity. If you take the muscle and do the EM, you can see this enlarged mitochondria here. This is almost 4 or 5 times bigger than the normal mitochondria. So clearly, high-fat diet induced abnormal mitochondrial accumulation, and we have this mitochondrial stress here, accumulation of red puncture, suggesting there is a possible a combination or maybe activation of mitophagy, maybe there is insufficient mitophagy, so this abnormal mitochondria accumulate in skeletal muscle. Now the question is, okay, high-fat diet induced abnormality. Can you show the opposite changes induced by exercise training? So I think this is the one that made us confident that this is the case. So here, it's 3 weeks of high-fat diet caused similar changes, not as severe, but similar changes in the FDB fibers. This is the 3 weeks of exercise training, did not show much of a difference compared to the normal sedentary mouse. But if you put them on a high-fat diet and subject them to voluntary wheel running, you don't see the abnormality. So this data suggests that not only our reporter can report pathology, but also in the impact of physiological exercise. So that's quantified here. Increased ratio, increased red puncture, and exercise training completely prevented or removed the damaged mitochondria.
Excuse me?
Yes?
So each of these is individual animals?
Each of those are individual animals. And the electron microscopy data also support this. So this is 4 weeks of exercise in the 12 weeks of high-fat diet fed animals. As you can see here, this abnormal mitochondria accumulated in the animal, and 4 weeks of exercise can significantly reduce the mitochondrial damage associated with the improved glucose intolerance. As you can see here, with a glucose tolerance test, this is the area on the curve.
One question. So, evidently, both exercise and normal diet, and the high-fat diet, sedentary state, both cases the mitochondria look really big and swollen. So, what are the structural differences that would make you think this is a healthy mitochondria that's bigger because of the exercise versus the non-healthy mitochondria?
So, exercise, we have in the literature, and we have found that exercise alone can only moderately increase the size of mitochondria. They probably improve the crystal structure of the mitochondria under the EM. With high-fat diet, the enlarged mitochondria always have this kind of disarray of the crystal structure. So exercise training may promote the removal of those damaged mitochondria. Because under those conditions, not only you probably have lipid overload to the mitochondria, which will possibly cause mitochondrial damage, mitophagy, but under the condition of high-fat diet, the arm for removal part is probably not activated, maybe even inhibited. So I think exercise has this benefit of not only put a stress on mitochondria, but also promote the removal of it. So our next question is this sufficient necessary for reducing high-fat diet? Just like Beth Levin's group, they did not look at the mitophagy, but they asked the question, can high-fat diet-induced insulin resistance be improved or exacerbated in the ATG6 genetic models? So we asked the same question, but with direct examination of the mitophagy process. So here is the plasma injection with overexpression of ATG6, which is sufficient to drive autophagy. And we inject Mito Timer with ATG6 construct and ask the question, can we see increased ratio on the condition of high-fat diet? Can that be mitigated by overexpression of ATG6? The answer is yes. As you can see here, here's the increase in the ratio with empty vector injection with a high-fat diet. But even under the high-fat diet condition, if you overexpress Becklin, you can mitigate the mitochondrial stress, suggesting that if you promote mitophagy, you can get rid of the bad ones that keep the healthy ones. The red puncta increased even in the absence of high-fat diet, suggesting ATG6 is sufficient to drive the fusion of lysosome with some suboptimal mitochondria, so promoting mitophagy. Then we subject those mice to isotope-based glucose uptake by insulin. So here is the glucose tolerance test. It appears to us that ATG6 heterozygous mice had an exacerbated glucose tolerance test with reduced whole body glucose clearance, reduced plantaris muscle, quadriceps muscle glucose clearance. And here is the glucose clearance in the brown fat in the heart. There are a trend of decreased glucose uptake in those tissues. So ATG6, which is an essential factor for autophagy, when you have reduced the expression, it reduces the capacity to clear mitochondrial damage, and that is associated with exacerbated glucose intolerance. However, when we use the Mito Timer to measure the change in ratios in the ATG6 heterozygous, we expected the result is exacerbation of more increase in the ratio or more increase in red puncta here induced by high-fat diet. We did not see that. This data suggests to me that our reporter gene may not be, I would say, quantitatively higher resolution enough for us to measure it. And so we need to really look into the analysis and to conduct more careful analysis. And plus, this is a plasma injection, so if we can generate a transgenic animal, which we are in the process of doing that, we may be able to improve the variability so that we can see more profound changes. So the take-home message I have here is that as a metabolically active tissue with a lot of mitochondria and mitochondrial function as an important, not only organelle function for muscle, for other cells, for the metabolism, for the function, but also as a kind of a sensor for the metabolic state, which could signal the cells for either stop the normal function or promote the function. So mitochondrial quality is important. Increased mitochondrial biogenesis through exercise, training, PGC1 is important, but we believe this clearance process is probably more important, as just like adding new cars to onto the traffic is important, but clear the dead ones, the clunkers, are probably going to be more cost-effective. Yes?
Just one step before your clearance, it seems you suggest here's the specific asymmetric fission process to sorting out damaging mitochondria. How much do we know about this process?
We don't know. This is hypothetical. This is hypothetical. But you pointed out, or we noticed that looks like the red puncta always getting very close to the network, So it could be possibly fizzed off. So it's not inconsistent with this hypothesis.
Do you see any difference before and after exercises, whether it's kind of asymmetric fission or if there's anything like that happening?
We haven't done enough experiment to look at that. You really need to get to the higher resolution and possibly, we are in the process of developing this ex vivo so we can culture those fibers in single fibers on the cover slip, and we're doing FRET imaging and we're doing time-lapse microscopy. Hopefully one day we really could address those questions with electrical stimulation, with change in medium, with a high-fat diet or palmitic acid. So that's the cash for Clunkers story. So I'm using the next few minutes to talk about 2 other projects, just a very brief introduction of that. So hopefully that will trigger some discussion, and I can certainly answer questions. So we have studied the muscle atrophy for many years, and we asked the question in the literature that there are a lot of reports.
Can I just ask a question up here before you continue?
Yes.
I was wondering if you could comment on, there's kind of this idea floating around that the benefits of exercise follow a bit of an inverted U curve, where if you get too much exercise, it's no longer beneficial for you, and potentially even detrimental. Is there any evidence that could relate that to like too much mitochondrial stress or something along those lines?
Yeah, I think philosophically that's always right. Too much is never good for anything. I don't think we have done any careful studies at this point to address too much exercise is good or bad. Yeah, so we haven't done anything like that. Yeah.
Great, thank you.
Thank you. So in this study, we asked an old question: why slow twitch muscle do not get atrophy? So if you search the literature, other than disuse, other than neuro problem, you don't get muscle atrophy in the slow twitch muscle. You get fast twitch muscle atrophy. Aging is another example, right? So we have found that with pathological insult like LPS injection or sepsis or dexamethasone injection, you have protection in the slow twitch muscle. Our question is, what are the possible mechanisms? And so naturally, we hone in on antioxidant, which I think is this school has a great expertise in doing that. So, and we found that the insults cause muscle atrophy actually induced antioxidant gene expression in slow twitch muscle far more than the fast twitch muscle. Those include SOD1, SOD2, SOD3, catalase. And then we conduct research, so we published those papers several years ago, and then we asked the question, can we use the possible regulator, we suspect that's an NO, and regulate these antioxidant genes? To our surprise, when we inject GSNO, which is an endogenous NO donor, we can readily induce EcSOD. This is SOD3 that can be secreted by cells. So in the literature, EcSOD has been reported to be highly expressed in smooth muscle and epithelial cells in the lung. So we asked the question, could that be induced in skeletal muscle? And we have lots of data confirm that skeletal muscle can express EcSOD. And exercise, a single bout of exercise increase EcSOD transcription, exercise training increase EcSOD expression in the skeletal muscle. So then we generated transgenic animal with a muscle-specific promoter. And in this study, we asked the question, can this transgenic animal be resistant to a condition like a heart failure? So here's the study. We subject these mice to a heart failure condition induced by genetic mutation, a genetic overexpression of a calcium handling protein in the heart. So this is like an intrinsic heart failure problem. And these mice developed the cachexia. You can see the body weight dropped significantly. When you put them on the treadmill, they don't run as much. And usually they run 3,000 meters. Here, they reduce to 700 meters. It's like a human patients, you ask them to do a 6-minute walk, they cannot walk the distance. And the gastrocnemius muscle get atrophy. But in the transgenic animal, the loss of body weight, loss of muscle weight, loss of capacity of running on the treadmill, all completely, significantly reduced. So this is the data suggests that overexpression of EcSOD in skeletal muscle is sufficient to protect the skeletal muscle from a condition that caused by the heart failure, which is a very important question in the cardiovascular research field. So then the question is this beneficial to other organs? And we notice that EcSOD get into the bloodstream in those transgenic animals. The level of EcSOD in the blood is 3 or 4 times higher than in the wild-type animal. And we found that all these tissues, organs, have higher level of EcSOD expression because it's coming from circulation. Because we know that the promoter is not active in any of those tissues. So we subject those mice to another disease condition called the type 1 diabetes-induced diabetic cardiomyopathy. We inject low-dose STZ for 5 days, wait at the end of it, only pick the animal with high blood glucose, and waited for 10 weeks. And look at the heart function. This is an echocardiogram and showing reduced ejection fraction, and that is blunted in the transgenic animal. We measure the fiber size, heart weight, and here only show the fiber size data. Clearly, there is a hypertrophy in the diabetic cardiomyopathy model, but blunted in the transgenic animal with the same level of high blood glucose. I have hyperglycemia. Fibrosis increased in the wild-type animal, attenuated significantly in the transgenic animal. So we have a hypothesis that the EcSOD overexpression in skeletal muscle can be redistributed to peripheral organs, tissues. And we have some evidence suggesting that EcSOD is structurally similar to copper-zinc SOD, but has 6 amino acid positively charged binding motif that allow EcSOD target to the cells such as endothelial cells, adipocytes. So it's like I call it a guided anti-ballistic missile. And so we are all excited about antioxidant, but we don't know how the endogenous antioxidant work. And here is the perfect example that our beautiful endogenous system is far better than all those drinks we can have, and which could destroy our microbiome, and it could target to the place we don't want them to target to. But this is an endogenous protein, knows where it targets to, and stay there waiting for the next insult. So today I do not have the data slide to show you. Actually, those mice are very resistant to LPS injection-induced endotoxemia. And if you fuse, I have the data to show that if you fuse the wild-type animal with a transgenic animal, the wild-type animal is now protected. If you fuse the wild-type animal, they don't have any protection. So this is very exciting to me as an exercise physiologist, that we believe this endogenous antioxidant genes can be induced by exercise training, can be redistributed to different organs, provide profound protection to those non-infectious conditions, those chronic inflammation-induced pathological conditions.
So, looks like this EcSOD crossed cell membranes. Is that correct?
Yes, there are studies showing that this can be internalized, but it's dependent on this heparin binding motif. So there's a human SNP showing that those patients with the mutation of the 6 amino acid. One of them mutated to G, from arginine to G, and it loses the ability to bind to heparin sulfate, and this molecule is circulating in the circulation without being able to target.
So presumably you could administer this, you could synthesize it.
Yeah, the problem with this is the large quantity of this endogenous produced protein that purified in ex vivo or in bacterial forms is not correctly modified. I think the quantity is not as good as skeletal muscle, which we can, it's 60% of the body weight, largest organ in the body. So this reserve in our body is huge. Yes?
Can it get into the brain?
We did that in the transgenic animal. We saw 5-fold change in different tissues, but in the brain, only very subtle change. But with the small sample size, we are not quite sure there is any get into the central nervous system. We did cross those mice with ALS mice, the one created here. They did not provide any protection. That was published in the paper.
Chen, one interesting experiment might be there are a bunch of EcSOD mimetics that are out there at the moment. One idea might be to covalently link it to your 6 amino acid targeting sequence and see whether it's actually effective.
That's a great idea. Yes, yeah. So next one is even far-fetched, and which is very different from this, but it's aligned with my research program in terms of exercise, mitochondria. This maternal exercise prevent parental offspring disease transmission. So when I was doing my research at Duke and I went to Singapore, I noticed that women in Asia do not have this large size. The obesity does not appear to be a major problem, but they do have the highest incidence of type 2 diabetes in the world, Singapore, okay? They don't get much obesity, but they have problems. And I noticed that ethnic groups there, Malays, Indian, Chinese, the cultural differences there is that they don't exercise as much, and particularly during pregnancy. So the question is, can that be a negative impact to their offspring, so it can be amplified? And whether that's true or not, I still don't know, but I can ask the question, can we ask mouse to exercise and mitigate the positive and negative impact of the parents? So we subject mice to high-fat diet, and half of them gave them running wheel and exercise. The offspring, we fed them with a normal chow for 9 months. And at this 3 months, 6 months, we did not see any differences. Body weight, no difference. Insulin glucose tolerance test, no difference. By 9 months, we saw a significant increase in the glucose intolerance in the not high-fat fed mice, the offspring from the high-fat fed dams. This increase was mitigated by maternal exercise. Interestingly, the PGC1 methylation, the reason we look at the PGC1 methylation, there was a previous study showing that exercise can decrease PGC1 methylation in the muscle of the individual who exercise, which is a direct impact of exercise. But we asked the question, can the mother exercise to induce hypomethylation of PGC1? Can high-fat diet induce hypermethylation of PGC1? The answer is yes. At the birth, the PGC1 methylation is higher in the offspring here, and it's mitigated by maternal exercise. It's sustained to the adulthood. At 9 months, the difference is obvious. And the PGC1 expression mirror that. It's like a reduced PGC1 expression with a hypermethylation of PGC1, increased PGC1 expression with a hypomethylation of PGC1. So not only that, PGC1 target genes showed same trend. This GLUTA4 expression is reduced, COX4, cytochrome C, all reduced in the offspring from the obese mom. And they all corrected, if not promoted to a higher level. In skeletal muscle only, not in the liver, not in the heart, not in those two tissues. So now we kind of formulated a new hypothesis and wrote a new grant that we are speculating that during maternal exercise, during pregnancy, it can mitigate the milieu-induced hypermethylation of PGC1 through this enzyme called DNMT3B, the DNA methyltransferase. And this methylation leads to the reduced PGC1 expression only occur during aging. So because we have the hypermethylation at the birth, and the mouse did not show phenotype. So there is an agent-associated insulin resistance which impinge on this PGC1. And so we can ask many different questions. Now our lab is conducting research with a paternal exercise, maternal exercise, paternal high-fat diet, with a muscle-specific DMT3B mutation, with a muscle-specific PGC1, transgenic PGC1 knockout to address those questions.
Isn't the confound weight? Yeah. I mean, so your exercised high-fat fed animals given exercise.
Yes.
What was their relative weight compared to the sedentary high-fat animals?
They are reduced. Their body weight reduced. This mom's body weight reduced moderately, not to the offspring. Offspring did not show much of a difference. Yeah.
How specific is this methylation? Is it global or PGC1 specific or promoting?
That's a great question. Whether this is a global hypermethylation or this is a PGC1 specific, we do not have the answer. And we really debated whether we should do the global approach, like a microarray type of approach, or method chip analysis, which allow us to look at every genes. In the end, we kind of decided to go directly to the PGC1 because the importance of PGC1 because of the previous finding. So you can do either way, but in the end, if you do the global one, you still have to ask the question, is the PGC1 important or not? So we kind of, cut the corner.
So you look at, methylation on the skeletal muscle. What about other tissues? Have you really looked at?
We did. We did.
What happened?
There was no increase or decrease in PGC1 methylation in the liver or in the heart.
What about other genes?
Other genes, we haven't. Yeah, yeah.
So this is methylation one side in the promoter.
In this case, we only focus on one side. There are reports of 3 other sites on PGC1 could be related to reduced expression. But in that previous study, in our study, this -261, which is this one, is the critical for PGC1 expression. So that has been linked to PGC1 expression. The other ones have not been linked to PGC1 expression.
Sorry, Ryan.
I'm just wanting to know how long would the animals exercise during pregnancy, and during what stage? Was it throughout pregnancy?
Yeah, in this study, throughout and even before the pregnancy. And in a recent study by Laurie Goodyear's group showing that it doesn't matter. It's during pregnancy or before pregnancy. If you go exercise, it can provide protection. So we have done a new study now. I don't have the data to show you, but I think the data strongly suggests that maternal exercise during pregnancy is just as powerful. It can mitigate the high-fat diet before, during pregnancy. Even the maternal negative impact can be mitigated by paternal obesity can be mitigated by maternal exercise. Yes?
So explain to me what I asked. You're going to look at placental function here, and that leads back to a question that I would have asked earlier in the first part of your lecture about the signaling, and in particular the relationship between signaling to mitochondrial amplification and brown fat.
Mm-hmm.
Since newborns are basically colonized with brown fat, what's going on there, and whether or not there's commonality in the signaling, what's going on?
Those are all great questions. Yeah, I wish we could get enough money and have enough people to work on those questions. And so there are, like this morning we had great conversation with Shelley's group and talking about the mole rats. And to my knowledge, this unique animal model exercise are a few of those encouraging models that really have a positive spin on all the things we study, right? So there are millions of questions, and so the question is which question we should go after, and to be right, number one, and to be funded, number two, in order to address those questions.
Because you would think there'd be some other signaling pathway that has the inverse here. We're looking at the stress models and signaling the placental function. And where you clearly have inducible reduction in placental function, and therefore, the whole energy supplies to the developing fetus.
Yes, yes, yeah. But the paternal high-fat diet data would suggest that it's probably earlier than that. Yeah, it's a very interesting. This is a new territory that I don't think I can move as fast as this question, which we can address. But the next questions are more difficult. Yeah, require different expertise. Yes. I would say I don't know. I would say I really don't know. The change in body weight by this type of exercise, and remember during pregnancy, this 19 days, they only exercise for half of that. So they naturally decrease their running distance to almost zero by the time they deliver the baby. So with like 10 days of exercise, we have not seen much of change in body weight. There is reduction in body weight, but not very dramatic. So if anything is not just the obesity per se, I would say something either induced by exercise or the impact of exercise have a direct impact on that process.
What strain of mice?
Those are black 6, C57 black 6 mice. All right, so I hope that I convinced you enough that exercise is good. Exercise is medicine. Exercise is the best preventive medicine. With that, I would like to thank people in my lab. I have a very active group, and they are all very young, and they go exercise. We don't just preach. We practice. We walk our walk. We run our run. And this individual run 100K. This individual, and many of those got a faculty position, moved on to their independent research, and they will make impact in the field about exercise and impact on health and the disease. I would like to thank my collaborators here, my lab members and the UVA collaborators. Jeff worked on this Mito Timer story with his postdoc, Kyle Han and Brandon Kingwood. I worked on the metabolic aspect, Lisa Palmer on the NO signal, Paul Adler worked on the Drosophila model, and I have a Jessica Connolly who's collaborating with me on the epigenetics. I have a lot of collaborators from outside of the university, and this is the beautiful campus of University of Virginia, which was founded by Thomas Jefferson almost 200 years ago. Thank you very much.